Hello everybody, welcome back to my Knowledge Academy channel. I am Mithu Dey. This is the part 3 video of the circulatory system. In my previous video, you have learned the process of destruction of RBC. Who have not seen this video till now, please go through the description box. Link is given in the description box, or you will type in the YouTube the Knowledge Academy by Mithu Dey. Without blood, the body's organ cannot get oxygen and the nutrients they need to survive. We cannot keep warm or cool off, fight infections, or get rid of our own waste products. Without enough blood, we would weaken and die. Here are the basics about the life-sustaining fluid called blood. If a person gets the wrong type of the blood, their immune system will react to it, and this is a serious condition that can cause the severe symptoms such as fever, muscle aches, and the respiratory distress, and so on. It can sometimes be fatal also. So we need to have enough knowledge regarding the blood, typing of the blood, grouping of the blood. So our today's topic are antigens and antibodies, ABO system and Rh factor, blood grouping, Rh incompatibility and Rh sensitization. So our today's topic is ABO system and Rh factor. So let's go. The discovery of the ABO blood group over 100 years ago caused a great excitement. Until then, all the blood has been assumed to be the same, and the often tragic consequences of the blood transfusion were not understood. At the beginning of the 20th century, an Austrian scientist, Karl Landsteiner, noted that RBC of the some individuals were agglutinated by the serum of other individuals. He made a note of the patterns of agglutination and showed that blood could be divided into the groups, and the discovery led to safe blood transfusion between people with compatible blood groups. This marked the discovery of the first blood group system, ABO, and earned the Landsteiner a Nobel Prize. What is ABO system? A system used to grouping of the human blood into the different types based on the presence or absence of certain markers on the surface of the RBC. Here, certain marker means the antigen A and antigen B. These are the certain markers. This ABO blood system is used to denote the presence of one, both, or neither of the A and B antigens on erythrocytes. That means grouping, ABO blood grouping, this system is depend on the what type of the antigen is present on the surface of the erythrocytes. So let's see the types of the blood group. Number 1, blood group A. Here A antigen is present. Blood group B. Here B antigen is present on the surface of the RBC. Blood group AB. Here on the surface of the RBC, A and B, two antigens are present on the surface of the RBC. And blood group O. Here no antigen is present on the surface of the RBC. Landsteiner called the antigen A and the B depending upon which antigen the RBC expressed. blood either belong to blood group A or the blood group B. A third blood group contained the RBC that reacted as they lack the properties A or the B. A and the B antigen here the lacks, and this group was later called the O group after the German word ohne, which means the without. The following year, the fourth blood group AB was added to the ABO blood system, and these RBC expressed both A and the B antigens. Let's see the antigen and antibodies. What is antigens? Antigen means any substances that causes the body to make an immune response against that substances. Antigen includes the toxins, chemicals, bacteria, viruses and other substances that come from outside of the body. Body tissue and the cells also have the antigens on them that can cause an immune response. Body tissue and the cells. Blood cells. We all know the blood composed of the plasma and the blood cells, and here the antigen is present on the blood cells. Antigens are the protein molecules and they are found on the surface of the RBCs. Here on the surface of the RBC, A antigen is present, so this blood group is the A, and here B antigen is present on this surface of the RBC, so this is the B blood group. And here on the surface of the RBC no antigen is present, so this blood group is the O blood group. And on the other hand, antibodies. Antibodies found always in the plasma. Mind that antigen always present on the surface of the RBC and antibodies always present in the plasma, and they denote the antigen denotes the capital A and capital B, and antibody denotes the small a or small b. Here these antibodies are the part of the body's natural defense and they recognize the foreign substances. That means they are the part of the body's natural defenses and they recognize the foreign substances such as the germs, that means the bacteria, virus, toxins, chemical, etc. And they alert the immune system which destroy them. Let's see the difference between the antigen and the antibodies. Antigens. Antigens are agglutinogens. Agglutinogens and the antigens are the same word, that means the synonyms that stimulate the production of the antibodies by causing the immunogenic reaction. That means agglutinogens or the antigens stimulate the antibody production by causing the immunogenic reaction. And antibodies are the agglutinins. Agglutinins and the antibodies are the same word, that means the synonyms, and they are capable of causing the agglutinations of a particular antigen. Agglutination, that means the clumping. Clumping of a particular antigen. Antigen, they are the different types of the antigens, maybe bacteria, maybe virus, or A antigen, B antigen, Rh antigen, different types of the antigens. And antibodies always depends on the host factors. So let's see the different type of the antigen and antibodies in the ABO blood group system. Pupil with blood group A will have A antigen, and as a result anti-A antibodies will not be produced by them because they would cause the destruction of their own blood. So they produce the anti-B antibodies. That means anti-B antibody will attack the blood cell that contain the B antigen. That means the blood group B. So RBC A antigen has the anti-B antibodies. Same as a pupil with type B blood group will have the B antigen, and as a result anti-B antibodies will not be produced by them because they would cause the destruction of their own blood. So they produce the anti-A antibodies. On the another way, we can say that anti-A antibody will attack the blood cell that contain the A antigen. So here RBC with antigen B means there is always present anti-A antibody, and person with AB blood group have the A and B antigen, but due to the presence of these two antigens they cannot prepare the no antibodies because they would cause the destruction of their own blood. And persons with O blood group, they have the no antigens, nor A or B. So they prepare both anti-A and the anti-B antibodies because there is no chance of the agglutination. How is the ABO blood type controlled by genetics? That means the genetic characteristics. Blood groups are inherited from both the parents, father and the mother. ABO gene is located on the chromosome 9 and has the 3 alleles A, B and O alleles. So what is the alleles? Alleles is the different forms of the same genes are called the alleles. For every gene has 2 of the same alleles. That means these are the same alleles. Or we can have 2 different alleles. Here it is the dominant alleles, it is the recessive alleles. Here A alleles and the B alleles are the codominants and O alleles is the recessive. So let's see the codominants and the dominants. What is the difference between the dominants and the codominants? Dominance is the situation in which the dominant allele is fully expressed while suppressing the recessive allelic effect of the phenotype. That means dominant allele is fully expressed over the recessive alleles. And next, the codominance. Codominance is the situation in which the both alleles work independently and express their effect in the phenotype without mixing the effect. That means both alleles are work independently and express their effect in the phenotype without mixing the effect of any one. Here A and the B alleles are the codominants. A and the B alleles are the codominants. That means the both alleles work independently and express their effect independently. That means the AB, they express independently and they work independently. And O alleles is the recessive. A recessive allele will only produce a phenotype if there is no dominant allele present. In other words, we need 2 copies of the recessive alleles in order to have the traits. That means 2 recessive alleles is needed in order to the trait. This table shows all of the possible combinations of the blood type alleles, and the blood type for each allele combination is shown in the right side of the table. And we have already known that each biological parents donate one of their 2 ABO alleles to their child. So let's see, if the father's allele is A and the mother's is A, so alleles will be the A plus A, so the blood group of the offspring will be A. And if the father's allele is A and the mother's allele is B, then alleles will be the A plus B, so the blood group of the offspring will be the AB. And if the father's allele is A and the mother's allele is O, so A plus O, alleles will be the A plus O, so the blood group of the offspring will be the A, because O is the recessive one. And BA is already explained, that means it will be the AB. And BB, if the father's allele is B and the mother's allele is B, then blood group of the offspring will be the B. And if the father's allele is B and the mother's allele is O, so B plus O, allele will be B plus O, so the blood group of the offspring will be B, because O is the recessive one. And if the father's allele is O and the mother's allele is O, so O plus O alleles, that means the blood group of the offspring will be the O, because 2 recessive alleles is needed in order to have the traits or the genotype. And OA plus OB is already explained, that it will be OA, it will be the A, and OB, that means the B. Let's see the various types of the antigens on the RBC. Besides A and B antigens, there are 50 types of the antigens on the RBC. Out of them, 5 are very important. 5 antigens are: number 1, antigen G, antigen capital C, antigen small c, and antigen capital E, and antigen small e. Remember that there is no small d. That means when we say the small d, actually there is no d at all. And these Rh antigens are also called the Rh factors. Among 5 antigens, antigen D are mostly prevalent. Antigen D, it is protein and strongly immunogenic, and it is mostly prevalent. Strongly immunogenic. What is the meaning of the immunogenic? Immunogenic means ability to provoke an immune response, and it is inherited protein. Why it is called the inherited? It is the inherited protein found on the surface of the RBC. Inherited means because it is passed from the parents to the child through the genes, and the fetus can inherit the Rh factor from the father or mother, that means the parents. Rh positive, Rh negative, and Rh null. When RBC contain Rhd antigen, that means here this green color indicate the Rh factor, that means Rhd antigen present on the surface of the RBC. So when RBC contain Rhd antigen, then he or she is the Rh positive. And when RBC does not contain Rhd antigen, then he or she is the Rh negative. Here this symbol, that is the black square, indicate the Rhd antigen, that means the Rh factors, and here these triangles, that is the yellow and the blue triangle, indicate the A antigen and the B antigen. So on the surface of these RBC has the A antigen and the B antigens, and also Rhd antigen. So this blood group is the AB blood group, but here D antigen is present, so it is the Rh positive, so AB positive blood group. And in the same manner, here A antigen is present on the surface of the RBC and also the D antigen is present on the surface of the RBC. So it is the blood group A blood group, but it is the positive because Rh D antigen is present here. So it is the A positive blood group. And so in the same manner it is the B positive and it is the O positive. Here A and B antigen, O means no A and B antigen. So here no A and B antigen, but here D antigen is present. So it is the O positive blood group. And here this blood group, this 4 blood group has no antigen D. So these are all the negative blood group. This is the AB negative, this is the A negative, B negative, and this is the O negative blood group. Here, when the RBC contain Rhd antigen, but these antigen molecules are not so densely, that means quantitatively defective, then it is called the weak D. That means here RBC with the normal expression of the D, this is the dense, but here decreased number of the D antigen. So it is called the weak D. And here when RBC contain Rhd antigen, but these antigen molecules are the defective in nature. That is the here this is the missing portions. So that means the qualitatively defective, then it is called the partial D. Rh positive is the most common blood. It is the most common blood type, and incidence of the Rh negatives is the 5 to 10%. And another type of the blood group, that is the null Rh. Null Rh. What is the meaning of the null Rh? It is the rarest type of the blood group, and here this is characterized by the lack of expression of all the Rh antigens. Here 5 Rh important Rh factor, that is the Rh antigens are the D, capital C, C, E, capital E, and the E. All the 5 antigens are also absent, and not only the 5 antigens, all the 50 antigens are absent. So it is called the Rh null. Researchers estimate that just 1 in 6 million people have the Rh null blood. Why is Rh null blood so precious? Besides making someone feels really special and valuable, as it lacks all the 50 possible antigens in the Rh system, this not only makes it rare, but this also means that it can be accepted by anyone with a rare blood type within the Rh system. This is why it is considered the golden blood. This is the golden blood group, and it is worth its weight in gold, as there is no antigen, there is no possibility of the agglutination. So it is considered the universal blood. Its life-saving capacity is the enormous. Is the Rh null the same as negative? This blood type is distinct from the Rh negative, since it has none of the Rh antigens at all. In the Rh negative blood group, only Rhd antigen is absent, but here this null antigen has no antigen at all. So it is called the golden blood, and it is also called the universal blood. We have already understood the ABO system and the Rh factor. On the basis of the ABO system and the Rh factor, we can differentiate the 8 types of the blood group. That are, at first we will discuss the negative blood group. That means Rhd antigen is absent on the surface of the RBC. So here it is the O negative blood group. It is the O negative blood group. That means here A and B antigen is absent and also antigen D is absent here. So it is the O negative blood group. And here A negative. A negative means only A antigen is present on the surface of the RBC, but Rhd antigen is absent here. So it is the A negative blood group. And it is the B negative blood group. B negative means only B antigen is present on the surface of the RBC, but antigen Rhd is absent here. So it is the B negative blood group. And here it is the AB negative. AB negative means A and B antigen are present on the surface of the RBC, but Rhd antigen is absent here. So it is the AB negative blood group. And these four blood groups are positive blood group. That means O positive. It is the O positive. That means here A and B antigen is absent, but positive means Rhd antigen is present here. Rhd antigen. This green color is the Rhd antigen. So only on the surface of the RBC, only Rhd antigen is present. So it is the O positive. And here it is the A positive blood group. That means A antigen is present here and Rhd antigen is also present on the surface of the RBC. So it is the A positive blood group. And it is the B positive blood group. B positive means B antigen is present here and also Rhd antigen is present here. So it is the B positive. And it is AB positive. AB positive means A and B, two antigens. A antigens and the B antigens, both antigens are present on the surface of the RBC. Not only the A and B antigens, but also Rhd antigen is present on the surface of the RBC. So it is the AB positive blood group. Identification of Rh factor is most important. But why? To determine the blood compatibility. That means to indicate whether the blood of the two different pupils is compatible when mixed, such as blood of the mother and her baby at birth. Pupils are either Rh positive or Rh negative. This distinction mostly matters when one is Rh negative and her child is Rh positive. Rh compatibility happens only when the father of the baby is Rh positive, and children may be Rh positive if they inherit the protein from their father, even if her mother is Rh negative. Next, the routine procedure. Checking for the blood compatibility is a routine procedure during the blood transfusion, blood donation and the pregnancy. The Rh factor is important in the blood donation and the transfusion. For example, an Rh negative person can only receive Rh negative blood. When people are given blood transfusion of the wrong blood group, the antibodies react with the incorrectly transfused blood groups, and as a result the erythrocyte clumps up and the stick together, causing them to agglutinate, and they may experience a hemolytic anemia. That means the hemolytic transfusion reaction, which can cause a serious complication. This is the hemolytic anemia. So this is the healthy baby and baby with the severe jaundice. What is the agglutination? Agglutination is defined as the formation of the clumps of cells or the inert particles by the specific antibodies to the surface antigenic compounds. That means after the reactions of the antigen and antibodies, the clumps of the cells or the inert particles occurs, that is the agglutination. Is blood agglutination good or bad? This is the agglutinated RBC in the circulation. The agglutinated RBC can clog the blood vessels and stop the circulation of the blood to the various parts of the body. That is very dangerous. And agglutinated RBC can also crack open and the leaking the toxic content out of the body, which can have the fatal consequence for the patient. So, blood agglutination is very dangerous for the health and it is also bad. What is Rh incompatibility? Rh incompatibility. Here mother is Rh negative and father is Rh positive. So, baby may be positive, may be negative. Rh incompatibility is a condition that develops when a pregnant woman has Rh negative blood and the baby in her womb has the Rh positive blood. Then mixing can occur, so Rh incompatibility happen. Cause of the Rh incompatibility. During pregnancy, RBC from the unborn baby can cross into the mother's blood through the placenta. This is one of the cause of Rh incompatibility. What is isoimmunization? Isoimmunization refers to the process by which a patient develops the antibodies to the cellular antigens derived from the another individuals. That means here the mother's blood is the Rh negative and the fetus blood, that means the baby's blood, is the Rh positive. So mother develop the antibody to the cellular antigen from the other individuals. That means the baby. The most common antigen involved with the isoimmunization is the Rh system, and isoimmunization is also called the alloimmunization. What is sensitization? A small number of the RBCs from the fetus circulation can cross the placenta and enters the mother's bloodstream. Anti-Rh D antibody can then develop in the mother's plasma in a process known as the sensitization. That means when from the fetus circulation small number of the RBC can cross the placenta and enters the mother's bloodstream, then only develop the anti-Rh D antibody in the mother's plasma. This process is known as the sensitization. So let's see the Rh factor sensitization. Mother is Rh negative and father is Rh positive and baby is Rh positive baby. This is the fetal circulation. This is a fetal site, fetal RBC, that is the Rh positive blood. And this is the maternal circulation, that is the Rh negative blood. This is the uterus and placenta separating from the uterus, and this is the placenta and this is the umbilical cord. So at the first pregnancy, during pregnancy, or the more commonly at the childbirth, a small amount of the fetal blood, small amount of the fetal blood, that is the Rh positive blood, enters to the mother circulation. Mother circulation after the next several weeks. This is the blood flow after pregnancy in the mother circulation. Over the next several weeks, the woman develops the antibodies. These Y separate are the antibodies. This is the anti-Rh antibodies. And immune memory develops against the Rh antigen. This is the Rh antigen. That means the Rh positive blood. So anti-Rh antibodies develops against the Rh antigens. And next, and the subsequent pregnancy, when the woman becomes pregnant with her second Rh positive child, her immune system quickly produces the antibodies and the memory, and develops the memory that attack the fetus red blood cells. So this is the anti-Rh antibodies. This is the anti-Rh antibodies, and these antibodies enters through the placenta, that means the umbilical cord, through the umbilical cord and the placenta to the fetus blood, and these anti-Rh antibodies attack the fetus red blood cells. So once again, why is the Rh incompatibility a problem? When the blood of the Rh positive fetus gets into the Rh negative woman, that means into the bloodstream of a Rh negative woman, her body will recognize that the Rh positive blood is not hers, and her body will try to destroy it by making the anti-Rh antibodies. And these antibodies can cross the placenta and attack the fetus blood cells, and these can lead to the serious health problems, even death for a fetus or a newborn. And when do Rh antibodies cause a problem? Health problems usually do not occur during an Rh negative woman's first pregnancy with an Rh positive fetus. This is because her body does not have a chance to develop a lot of the antibodies. But if treatment is not given during the first pregnancy and the woman later gets pregnant again with an Rh positive fetus, she can make the more antibodies, and more antibodies put a future fetus at risk. Can problems be prevented? Yes. The goal of the treatment is to stop from making the Rh antibodies, and identification of the Rh negative early in the pregnancy or before the pregnancy, that is the blood grouping and the cross matching, blood grouping and typing. Negative blood identification is the first and foremost things. And next, the medication to prevent the antibody from the forming. Can Rh antibodies develop when a pregnancy is not carried to term? Yes. An Rh negative woman can also make an antibodies after the miscarriage, ectopic pregnancy and induced abortion. What is Rh immunoglobulin? Rh immunoglobulin is a medication. It is a medication. Anti-D immunoglobulin, that means the Rh. It is for the intramuscular administration that stops the body from making the antibodies. That stops the body from making the antibodies and prevents the severe fetal anemia in a future pregnancy. And Rh immunoglobulin is given as an injection form. That means the intramuscular injection only. it is not helpful if your body has already made Rh antibodies. When would I need to take Rh immunoglobulin? At 28 weeks of the pregnancy. A small number of the Rh negative women may be exposed to the Rh positive blood cells from the fetus in the last few months of the pregnancy and may make the antibodies against these cells. And Rh immunoglobulin given at the 28 weeks of the pregnancy destroy these Rh positive cells in the woman's body, and this prevents the Rh positive antibodies from being made. And next, the within 72 hours after the delivery of an Rh positive baby, the greatest chance of the mixing during the delivery. That means the greatest chance that the blood of the Rh positive fetus will enter the bloodstream of an Rh negative woman occurs during the delivery. And Rh immunoglobulin prevents an Rh negative woman from making the antibodies that could affect a future pregnancy. And the treatment is good only for the pregnancy for which it is given, and each pregnancy and the delivery of an Rh positive baby requires a repeat dose of the Rh immunoglobulin. So, at a glance we can say that Karl Landsteiner discovered ABO system and Rh system. According to the antigen A and the antigen B on the surface of the RBC, blood can be classified as the 4 groups A, B, AB and O. And also antigen D, on the presence or absence of the antigen D on the surface of the RBC, we can classify blood as the 8 major groups: A positive, A negative, B positive and B negative, AB positive and AB negative, and O positive and O negative. And also Rh incompatibility and the sensitization, that is, as a result of the incompatibility of the Rh positive and the Rh negative, the anti-Rhd antibodies develop in the mother's blood. So the fatal conditions of the babies occurs. So we need to take care of our mothers and also the baby. Thank you.